Hello everyone, good morning to you guys. Hope you are all doing so well. Okay, so this is your wonderful tutors, Kenyam Voyas, and I'm here for us to discuss about a wonderful process in tissue respiration, which we call glycolysis. So, what is glycolysis? Advanced level biology. What is glycolysis? 10 marks. 10 marks. So what will you write for glycolysis, dear students? What will you write for glycolysis to get to score 10 marks? So you may begin by recalling what is tissue respiration, which I don't really think, which will not really make part of the answer, but at least. So glycolysis is the first stage. You begin by telling the examiner that glycolysis is a first stage of tissue respiration. And tissue respiration is the enzymatic breakdown of energy-rich substrates like glucose, fats, right? But generally, fats are not broken. Sometimes fats actually enter the Krebs cycle directly. So tissue respiration, or to get back, glycolysis is a first stage of tissue respiration, which and tissue respiration is the enzymatic breakdown of energy-rich substrates like glucose, with or without oxygen, with the objective of producing energy in the form of ATP. When there's oxygen, tissue respiration is called anaerobic respiration, and when there's no oxygen, it is called anaerobic respiration. Whether aerobic or anaerobic, glycolysis is a first stage of all forms of tissue respiration. And glycolysis simply is a breakdown of glucose. Glycolysis, therefore, is a series of chemical reactions, by which glucose is broken down to produce pyruvate. Glucose is broken down to produce pyruvate as the intermediate in the process of producing ATP from glucose. Glycolysis occurs in the cytoplasm of cells. It occurs in the cytoplasm of cells and has glucose as its input, or its substrate, or its major input. The process of glycolysis uses glucose, uses ATP, and uses oxidized NAD or NAD+, and produces as output, it produces ATP again, right? It produces more ATP, it produces pyruvate, and again, it produces reduced NAD. So the inputs of glycolysis are ATP, glucose, and oxidized NAD or NAD+, while the outputs are ATP again, pyruvate, and reduced NAD. Glycolysis begins with glycolysis can be broken down into two stages. The first stage involves the double activation of glucose, and the second stage involves the lysis or the breakdown of the glucose. So glucose is first activated by oxidative phosphorylation. Phosphorylation simply means the addition of a phosphate group to any compound. So glucose, from glucose which you get from the food we eat, is first oxidatively phosphorylated. Oxidative means that as a phosphate group is added to glucose from ATP, right? ATP adds phosphate group to glucose. At the same time, reduced NAD, oxidized NAD+, removes hydrogen from glucose. So the first oxidative phosphorylation of glucose produces a compound that we call glucose 6-phosphate. Glucose 6-phosphate will undergo a structural rearrangement, right? To form fructose 6-phosphate, which would then be oxidatively phosphorylated once again in the second part of the activation to produce fructose 1,6 biphosphate. At this level, the glucose molecule has become unstable and can easily break down. So we now dive into the second reaction of, or the second stage of glycolysis, which involves the breakdown of fructose 1,6 biphosphate into two molecules of pyruvate. Glucose is a 6-carbon compound, and when broken down, it breaks down into two identical 3-carbon compounds, which we call pyruvate. So fructose 1,6 biphosphate then undergoes a series of reactions which involves first and second levels substrate phosphorylation, right? And the production of molecules of ATP. This levels phosphorylation eventually convert fructose 1,6 biphosphate into two identical molecules of glyceraldehyde 3-phosphate, and which is hydrolyzed to produce phosphoenol pyruvate, and phosphoenol pyruvate further is converted into pyruvate. So that is the series of reactions by which glucose is converted to pyruvate in the process of glycolysis. The inputs, as we earlier said, were glucose and two molecules of ATP, as well as two molecules of oxidized NAD. The outputs are pyruvate, two molecules of pyruvate, right? Because fructose 1,6 biphosphate was broken down into two parts, so it gives us two molecules of pyruvate. Now, for each pyruvate molecule, there were two levels of there were the production of two molecules of ATP. So overall, glycolysis produces 4 molecules of ATP, and the net output is 2 molecules of ATP, because in the initial stage of activation, the process used up 2 molecules of ATP. So it produced 4 ATP and uses 2. Overall, it has produced 2. So what happens now to pyruvate is what we call the fate of pyruvate in tissue respiration. If there is oxygen present, then pyruvate will enter the Krebs cycle, right, where it will undergo a series of reactions to produce 2 more ATP, right, as well as carbon dioxide in a process of decarboxylation. Pyruvate from the Krebs cycle will then enter the electron transport system, where more ATP, abundant ATP of up to 34 molecules are going to be formed in the electron transport system, which is the last stage of tissue of aerobic respiration. But if there's no oxygen, then pyruvate will have to take one of 3 possible anaerobic pathways. One of the first pathway is what we call alcoholic fermentation, where pyruvate will be oxidized into ethanol and the water. The next pathway is what we call lactic acid fermentation, which occurs in human muscle cells. There, pyruvate will be converted to lactic acid without water and without carbon dioxide. The next alcoholic pathway is when the next fermentation pathway is when pyruvate is converted to acetic acid in some bacteria that we call in some bacteria that belong to the genus Acetobacter. So these bacteria are able to convert the ethanol, which was produced from alcoholic fermentation, into acetic acid by first of all converting it to ethanol and hydrolyzing it into ethanol hydrolyzed ethanol, then reducing it to acid, oxidizing it to acetic acid. So the whole of any fermentation pathway is mainly to produce back or to replenish oxidized NAD, because glycolysis uses oxidized NAD to produce reduced NAD, right? So, and for glycolysis to continue taking place, the oxidized nut which was used in the process has to be produced back, and that is the main goal of all the three fermentation pathways, whether it is alcoholic fermentation, whether it is lactic acid fermentation, and whether it is acetic acid fermentation. So that is the wonderful process of glycolysis, and that is the fate of pyruvate at the end of glycolysis. Thank you, dear students. Make sure you like the video, subscribe to the channel, because more lessons will be coming up. Have a wonderful day and keep studying very hard for yourself, for your families, and for PDV LEADEX education. We are in various centers in Yaoundé and in Douala, in Essos, in Etukebe, in Mendong. And more centers will be coming up to spread to the different corners of Cameroon, as well as beyond. As well as in Douala, you can find us at Bobongo, you can find us at Ngodiari, you can find us in small other centers which will be coming up. Have a wonderful day and God bless you. Thank you.